Morning, everyone. Thank you for being here, sticking around Sunday morning for this session. So this is Pharmacovigilance, Follow the Evidence. We have 6 great speakers here today. And yeah, we'll just get right started. We're going to try and stick to the schedule so everyone can get the time to present and follow up with questions. So to start off, I'll give the floor to Christopher Gravel, who is from the University of Ottawa.
Okay, good morning, everyone. So my name is Chris Gravel. This is joint work with my colleague Antonius Duros, and I'm going to talk a bit about the use of different reference sets in data mining spontaneous reports for pharmacovigilance. Okay, so a quick summary of my talk. I'm going to start off by introducing spontaneous reporting and disproportionality analysis for drug safety signal detection, talk a bit about how we choose reference or comparator sets, and how that reference comparator set restriction in data mining has an intended impact on bias. Then I'll talk a bit about how the influence of spontaneous reporting may impact the intended bias reduction mechanisms of comparator set restriction. And then I'll demonstrate this with 2 empirical examples using the FDA's adverse event reporting system data, specifically canagliflozin in acute kidney injury and rivaroxaban in hepatotoxicity. Okay, so spontaneous reports are unsolicited communications by a healthcare professional or consumer. They describe suspected adverse drug reactions. They may contain limited covariate information, and sampling depends on the decision to report. So I placed a hypothetical DAG here where you can sort of envision somebody who takes a drug, experiences a suspected adverse drug reaction, and that motivates this reporting decision. However, it also depends on measured or unmeasured variables, which may end up being potential confounders of the drug event relationship. And it also may depend on the drug itself. In a given time frame, we get hundreds of thousands of these reports. So we get loads of medication and adverse events. So we can represent these data as sparse multidimensional contingency tables, which can be collapsed for analysis. So this data generating process induces intractable biases, rendering measures of association inappropriate. In addition, it's denominatorless, and there's insufficient available information to recover consistent estimates of the drug event combination risk. So instead, what we tend to do is estimate disproportionate reporting rates, where we compare the reporting rates of the drug event combination of interest against the reporting rates of the event within some comparator set. And the assumption here is that heightened disproportionate reporting is a proxy for actual risk, which can then generate a drug safety hypothesis for follow-up and alternate data sources. So the crux of this talk is about how we select these comparator drugs and what impact they may have on bias. And so to introduce this, I'm only going to demonstrate this with 3 different reference sets. There's many more in the literature, but this is just for demonstrative purposes. So the first reference set is the full comparator set. This would be all other drugs in the spontaneous reporting database. And there's no intended bias reduction mechanism here because it's literally the full representation of the observed data. And so I'm going to call this the benchmark set, since this is sort of like there's nothing sort of impeding this data set. And for the canagliflozin AKI example, this would be a comparison of canagliflozin against non-canagliflozin SGLT2 inhibitors, DP4 inhibitors, and all other drugs in the database. The next set is the active comparator set. And given that this is a pharmacoepic conference, I'm sure many of you are familiar with the active comparator new user design from pharmacoepidemiology. But with spontaneous reports, we have no information on drug utilization patterns. So we cannot implement the new user aspect of this design. Instead, we can just select active comparator drugs. So these would be drugs with similar indication and without contraindications. And the intended bias reduction mechanism here would be confounding by indication and channeling. And the third one I'm going to demonstrate takes the active comparator set and also excludes drugs from the same class. So we've called it the active comparator class exclusion set. And the additional bias it's attempting to address is called the confounding by association, or the innocent bystander effect, where if you have multiple drugs within a spontaneous report and you have regular co-prescription with drugs in the same class, you might get a spurious signal simply due to the co-prescribed drug being the drug that's problematic. So this set would be the canagliflozin versus DPP-4 inhibitor direct comparison. Okay, so the major question I'm asking today is, what is the influence of spontaneous reporting on the intended bias reduction mechanisms of comparator set restriction? So on the first slide, I showed you this very biased DAG without sufficient information to recover consistent estimates. And the question is, how does confounded outcome-dependent sampling filter through a reporting probability end up altering that sort of confounded relationship? And will it preserve the bias reduction mechanisms intended by these study designs? And so to think about this a little, we can think about what does the reporting probability depend on? Well, we know it depends on population outcome prevalence, it's outcome-dependent sampling. We also know it depends on clinical and demographic characteristics of the reporter, but it also depends on external events that may be related to adverse reactions or the drugs of interest, such as calendar time-dependent factors, the history of the drug product life cycle age, notoriety, and so on. So to take a look at this in more detail and to understand a bit about how this potential reporting bias or how these reporting biases may alter the ability to use these active comparator and active comparator class exclusion sets, we'll consider two drug event combinations, which can be described as null effects based on randomized trials and pharmacoepi studies. So the first is the canagliflozin acute kidney injury drug event combination I've already described. And in the literature, it's been compared against DPP-4 inhibitors. And rivaroxaban and hepatotoxicity has been compared against vitamin K antagonists. But the other reason why we picked these particular combinations is because there are potential external causes for enhanced reporting in their history. So in 2016, the FDA issued a warning on canagliflozin and acute kidney injury. And for the rivaroxaban combination, there was a previous oral anticoagulant therapy that had a confirmed hepatotoxic risk prior to rivaroxaban's market authorization, which may have induced a Weber effect. And for those of you who are unaware of the Weber effect, there's a hypothesis that the first 2 years post-market authorization stimulated reporting occurs, which has been shown on and off depending on the drug event combination. And so these two drug event pairs will hopefully allow us to see a bit about how the reporting bias interacts with these restricted reference sets. Okay, so the first one, canagliflozin acute kidney injury. So on this plot, this is a forest plot in 2 time intervals. The upper plot is market authorization till just before the FDA warning. The bottom plot is market authorization to 2022. This is all done in, the FDA's adverse event reporting system data, by the way. And you can see on the y-axis, I have the full data or the benchmark set at the top, followed by the active comparator, and then the active comparator class exclusion. And there are 3 confidence intervals. I made the benchmark in red, and the 2 restricted reference sets are presented in black. And these are all on the log PRR scale, or the proportional reporting ratio. For those of you who are unaware, that would be the analog to the risk ratio in spontaneous reporting data. And so what you can see here is that the canagliflozin acute kidney injury drug event pair produces a signal. However, the benchmark or full data comparison set has the largest signal, and the active comparator and active comparator exclusion set attenuates the signal towards the null in both time intervals. So this appears to be working as intended. However, for rivaroxaban and hepatotoxicity, the opposite is true. So in this plot, we start at 2011, which was market authorization, and then I've done 2-year intervals extending the window of observation across the 4 plots. And so the first upper plot would represent the period of time where the Weber effect may have occurred. And you can see that a signal has been detected again, but the benchmark or full data reference set has the most attenuation to the null. And then over time, the signal will dissipate towards the null until it crosses somewhere in and around 2016 for the full data comparison set, whereas the other 2 would maintain a signal for longer. So this demonstrates that there may be some problems with these restricted reference sets relative to benchmark, and benchmark again is all drugs in the spontaneous reporting data. So in summary, reporting bias may distort intended systematic bias reduction mechanisms based on study design approaches. the use of restricted comparator sets does not necessarily guarantee less biased estimates. I also ran the Bayesian methodologies, the BCPNN and EVGM for those who are aware of the field. Only minor differences were observed, and the EVGM also did not converge in a lot of the cases because of how much smaller the set of drugs in adverse events are. So it had problems with convergence. And then finally, more research is needed to clarify whether reference sets may be consistently applied under specific settings. So for example, within a particular drug class or indication and so on. And that's it. Thank you. Are there any questions?
Thanks for a great presentation. Stuart Geary with ASI. There's one other fundamental problem with all the disproportionality signaling databases which use, for instance, the FDA FAIRS database, and that is the reporting differences. So for instance, the FDA requires expedited reporting of serious reports from the US if they're expected or unexpected. So all those will get in. But the foreign reports will only get in if they're unexpected. And the other problem is whether the case is going to be classified as serious or non-serious. And AKI is a kind of event that sometimes be non-serious and serious. What are your thoughts about how to control for that variability? Because it can be quite substantial depending upon whether the drug is marketed a lot outside the US and it's got a lot of cases which are not getting there vis-a-vis the other cases, the other drugs in the database.
Right. So I think this is another example of how this comes into the reporting bias, into that DAG I demonstrated, right? We're only able to see the drugs that end up obviously into the database. And the question is, what does the non-observed set look like? So if you're stratifying by different country and you're getting different style, like types of reports in, you'll have even worse bias, and you'll have an even harder time telling whether these bias reduction techniques are working as intended. But you may potentially look at just within the US or within specific locations to determine if you can see differences between the reporting, let's say the reporting rates if you stratify by country, and you might be able to get some information from there.
This analysis is taking all the cases in the database, not those from the US originally or from it's not ignoring those of which were foreign reports.
So to clarify, I'm not suggesting you should only use the full data reference set. I'm saying this is just to demonstrate that you cannot. So in the canagliflozin example, the full data set would have been the worst choice. In the rivaroxaban example, I hate to say the best, it would have been a better choice. But I think all the choices are biased, and that's sort of the crux of the talk is no matter what you do, you're incurring bias, and so you kind of want to really think and understand about what you're doing when you do a restricted reference set, and perhaps do multiple since it's data mining and it's, you can rerun the analysis a few times to triangulate the signal.
Thank you.
Thanks. Anyone else?
Maybe I have one comment. Can you elaborate on potentially the different forms of biases, or is it the same form? You're saying because all the different approaches have bias, right?
Right. So I guess so. There's been some great talks throughout the conference on different biases. There was one yesterday that went through and characterized a lot of the pharmacoepi related biases. So the ones that we're all used to, confounding and mortal time bias, like all of these types of biases. So we write these DAGs, we try to create identification conditions, and then put it through this filter. And then this reporting probability will sort of make it so that they don't work as intended. And so some of them are, some people call masking or competition bias, where you may have a signal that's blown up for some external reason that muffles other signals. There's, confounding by association, the innocent bystander effect, notoriety bias, also a drug that has a lot of time in the press might induce in heightened reporting. So there's a lot of these external types of biases in pharmacovigilance or in spontaneous reporting data, I should say, which may alter the sort of traditional epi biases.
That's great. Thank you. Yeah. Okay, if there's no further questions, we'll go to our next speaker, Eva Lisa Meldo from the Uppsala Monitoring Center.
Good morning. Since you all got up early, despite the long conference days and the social event last night, I guess you all care about pharmacovigilance. And I also bet you're a bit tired. My name is Eva-Lisa Melder, and I am passionate about method development for pharmacovigilance. And I'm definitely a bit tired, but I'm also excited to talk to you about some of the work that we've done on medication errors related to high doses using outlier detection. So let's see if I can keep you awake. My disclosure, I have nothing to disclose. And yeah, so the other day I was standing at the kitchen sink, cleaning strawberries, distributing them into bowls. And I was wondering which of the strawberries might be the most tasty ones. So I thought back on what do normal strawberries, good strawberries look like my experience of what is a good strawberry. And I looked at the ones I had one by one and thought about what's the color? Are they like brighter or darker than the good ones in my experience? Are they maybe bigger than normal ones and therefore maybe watery? So if you look at the ones in the middle, do you think, raise your hand if you think they would be tasty? Come on. All right, and who of you thinks is a bit skeptical? Yeah. So I actually haven't tried these ones. I took the image of the internet, so I can't tell you if they were tasty or not. But yes. So prescriptions of medications above recommended dosages is one of the most common medication errors responsible for causing preventable harm. And when I say medication errors, what I mean is unintended failure in the treatment process that leads to or has the potential to lead to harm to the patient. So this failure could, for example, be during prescribing, but also dispensing and administration by the health professional, or it could also be by the patient. And it's good to note that some of the cases of off-label use with high doses can actually be rational use, so where there's a reason to prescribe a high dose to the patient. But what we were interested in is the ones where we have a high dose, but they were not rational use. And the information about the rationale when we have adverse event reports can be often found can be only found in the case narrative description on the adverse event report, but actually turned out to often be missing in our explorations, which I'll come to later. It's a bit challenging. So what we wanted to do was to know if we can actually perform quantitative signal detection on reports where a patient have taken a high dose of a drug. So dose that is higher than the recommended daily dose. And what we were looking for was systematic errors. So something that happened more often where maybe also we could see the same root cause multiple times. So maybe the label would be unclear. So healthcare professionals prescribers would prescribe too high doses, or maybe patients would misunderstand the instructions. So we also just wanted to focus on reports which had been not coded as medication errors. So you could give an adverse event report and MEDRA code for an adverse event code saying that it is a medication error, something like incorrect dose administered, but those you could capture just using those codes. So that's actually something that colleagues of mine have already done, and they've successfully identified medication error signals that way. So we were interested in the uncoded ones. And yes, the problem with knowing what is a high dose, apart from the difficulties that come from the complexity around what's high for an individual, depending on characteristics like age and weight, is actually, we wanted to know just what's high in general. And that's already difficult because there's no comprehensive and, most importantly, easily machine readable data source that will tell you all the recommended daily doses for all drugs in the world. So you have the label, but that's free text. So if you want the daily dose, you would actually have to find that automatically in all the labels, extracted to get that information. So if you think back to the strawberry example, what we wanted to do is use our experience. And in our case, that is the VigiBase, the WHO global database of adverse event report, which we maintain at the Uppsala Monitoring Center in Uppsala, Sweden. So in this project, we decided to see if we can use the reports from Vigibase to identify the commonly reported daily doses and see if we can then identify outliers to see cases of high dose. So we use statistical outlier detection in form of the robust z-score. So we looked at the distribution of the daily dose, which could look something like this, and then we would look for the median dose, and also the difference of each reported dose to that median. And based on that, we can compute this z-score and for each of the reported drugs and identify those which were higher than a certain threshold. And this threshold we computed on some selected drugs before. So by using this z-score method, what we could do is compute one threshold that would work for, or we hope would work for different drugs, and different dose distributions. So we wouldn't have to say for this drug, everything from 200 is high, but actually anything that has a certain distance from the median, and which is appropriate for that underlying distribution is higher. So given adverse event reports from Vigibase, we could use the reported doses on the report to calculate the daily doses, because of course they're not given in daily doses, there's a lot of fields, so you can compute that. And then we use this z-score to identify outliers. And as I said, we focused on reports that were not already coded as medication errors. And we then identified drug adverse event combinations and their high dose reports, and checked if we could, based on that, identify medication error signals. And of course, we couldn't look at all combinations that we could find, so we needed to prioritize. So we prioritized based on the drug adverse event combination based on the odds of seeing the adverse event at a high dose as compared to a normal or low dose. So equipped with this approach, we came together for a signal detection workshop with participants both from Uppsala Monitoring Center, but also from the Moroccan Pharmacovigilance Center. So here you can actually see all the co-authors. We clinically evaluated, identify high dose reports for 62 combinations, and that we identified from this ordered list that were ordered by this odds ratio. And for these combinations consist of 898 reports, of which only 56 had a case narrative. So remember, I mentioned the narrative has often or could have the rationale, but if the narrative is already missing in Vigibase, we wouldn't know the rationale for those ones. And then when looking at the combinations manually during this manual clinical evaluation, we found that 11 of the, so we also could then compared the doses that we actually saw that we picked up as outlying doses, compared them to the label to see were they actually above the recommended dose. For 11 of the combinations, we actually found that they were outlying, but they were not high if you looked at the label. All of them. For 25, we found there were both those that were above and those that were high, but not above the threshold, and according to the label. And for 26, all of them were actually above these the recommended daily dose. So as you can see, maybe the threshold doesn't generalize perfectly, but we still had very interesting combinations to look at. So this still helped us to identify interesting combinations to look at. But what we ran into next, the problem that we ran into next was this problem about the rationale. So sometimes we didn't have the narratives, but even when we had the narratives, reporters were not really reflecting on why they were giving these doses. They would write narratives for reporting adverse events. So there was a lot of reflection on causality maybe, but not so much about why was this dose chosen. So yeah, we actually couldn't identify any root cause, and yeah, it was hard to actually identify signals, or impossible. So to sum it up, what we found was that pharmacovigilance has the potential to address medication errors related to high dose, for example, those related to high doses. And it's actually possible to use statistical outlier detection such as the z-score method to identify those outlying dose reports, high dose reports, without this external reference set. So we didn't need to have a set that would tell us all the recommended daily doses, but we could still identify some high dose cases. But if we want to do signal detection, we need more reflection during reporting about the doses and medication errors in general on spontaneous reports. With this, I would like to thank you for your attention and hope that you will start thinking about how you can maybe improve and increase the reporting of dose information on adverse event reports. It's not always given, I can tell you. To improve the reflection around medication errors and also doses during reporting, and how you could use the already existing dose information that exists on adverse event reports for medication error signal detection. Thank you.
Highlights kind of the need for us to kind of update our thinking about the kind of the standard reporting form in the era of electronic medical records. And has the UMC started to think about perhaps to, you know, I don't know, and I'm not a IT person, so but presumably in the electronic medical record world, there must be some standards, because at least in places like US, people change health insurances almost on an annual basis. So the records hopefully can be translatable in some way, and one could potentially just tag a record in some way.
That's an interesting idea. So we're quite far away from the electronic records, since the way we receive reports is they first get sent to, for example, the FDA or the MPA in Sweden, and then they come to us. So we've mostly been focusing on the adverse event reporting after it's been at the regulator or the national center. I can say that we've thought a bit about, could we use this method, for example, during reporting of adverse event reports? And I guess that would translate to electronic health records as well. If you would always show, I mean, I guess it's already done during prescription that you have these alerts, but can we have these kind of alerts of this is high, you know, even during reporting, just to ask these questions, not to everyone reporting an adverse event, but maybe the ones who seem to be reporting a dose that seems to be high. Can we like trigger them to reflect on it by asking the right people those questions, so that not every report you fill out you have to reflect on the dose, but the cases where it seems to be deviating between, like diverging from common practice.
Tarek Ahmed from Takeda. I'm wondering, did you think about linking some of those reports identified as above those that are supposed to be given with actual other clinical adverse events? So not maybe you'll find some association. This can strengthen your approach that you are identifying some consequential increase in the doses, not just an increase.
Sorry, can you repeat the last sentence?
So this can help you maybe make the case for your approach that can help you identify consequential increases in the dose, because they are leading to actual clinical adverse events.
Mm. No, I don't think we've thought about that, but we definitely, thought about focusing on the things that actually lead to harm. So it was one of the reasons why we focused on the adverse event. But I don't think I answered your question, because I don't think we've discussed that, not as far as I know.
Okay, thank you so much, Eva-Lisa.
Thank you.
Our next speaker will be Aziz Shobi from Johnson & Johnson.
Okay. Good morning, everyone. Now that Eva-Lisa woke us all up, thank you for that. As a true epidemiologist, I'm going to take you away from spontaneous report back to observational data. My name is Azza Shoaibi, and I work for Janssen Pharmaceutics. I'm going to talk to you today about the development and evaluation of phenotype algorithms for the European Medical Agency, EMA, designated medical events, DMEs, in a network of real-world data. I worked with this with a group of collaborators, where we are all employees of Janssen, and here are our disclosures. Okay. For those who are not familiar, DMEs is a list of MedDRA preferred terms that the EMA uses to identify spontaneous reports that are worth immediate and special attention because those believed to signify clinical events or adverse events that are rare but serious and often causally associated with the use of drugs across multiple pharmacological and therapeutic areas. It's a long list, but here are some examples: acute hepatic failure, acute kidney injury, and so forth. So what we wanted to do is we wanted to develop and evaluate phenotypes to identify DMEs in observational or real-world data, such as electronic medical records and EHR. We want to do this in Janssen because we are working hard to develop and implement a proactive safety surveillance system that is able to detect signals using real-world data. So of course, the first thing you need to do is if you want to detect something, you need to define it and know how to capture it in your data sources. So we followed a 7-steps process, and while it looks nice and linear on the slide, it wasn't at all. It's a bit of an iterative process, but we start with grouping those MedDRA PTs, which sometimes are super granular, into 32 clinical clearly defined outcomes with clinical descriptions and clinical definitions from our clinical teams. We then ran a systematic literature review to try to identify validated algorithms existing already in the public literature or maybe code lists. We then developed phenotype algorithm using Odyssey tool Atlas because all our data sources are mapped into the common data model, OMAP common data model. We also then took this algorithm, implemented them across 10 different observational data sources, and then we characterized the patients who met the definitions for those algorithms. We then did a review of the group level characteristics of the people who met the definitions across the DMEs, and I'll tell you later how was that useful. Then we selected a random sample of cases for people who met the definition, and we went what we call patient reviews. Patient profile reviews. Those are not case adjudication. We do not have any ability to access the full records because this is all de-identified data. But what we did here is that we looked at the structured data available in the electronic medical records or claims for a set of cases. And then we calculated the operating characteristics, or we estimated the measurement error for those phenotype algorithms using a probabilistic model as a gold standard. So let me walk you through acute hepatic failure, which was a not easy example of how we did those 7 steps. So on the DME, you'll find acute hepatic failure, and hepatic fulminant. Our clinical team asserted that the clinical event, the outcome we're interested in here should be acute hepatic failure in the context of drug safety. They also provided a clinical definition and description, and here they use an authoritative case for each of the DME to come up with a consensus of what clinically the case we're interested in. Then we did the second step of our systematic review, and in acute hepatic failure, actually, there's a body of literature for studies who try to identify acute hepatic failure in electronic records or EHRs or claims. And here I'm highlighting 2 papers, one that came out of Sentinel. And that all happened in the world of ICD-9. And this is particularly important because acute hepatic failure actually doesn't have a specific code in the ICD-9 vocabulary. So those papers suggested some proxies, but they also reported that the PPV, or the measurement error, or the positive predictive value was pretty high, or pretty low for a PPV, somewhere between 5 to 20%, which is really awful. That means only 5% of the people you're actually detecting truly have the case. So we took what we learned from literature review and what we and the clinical definition that we're interested in, and we tried to model that in observational data. And we often end up with more than one phenotype algorithm that we want to evaluate. So for acute hepatic failure, this is one example of a phenotype algorithm. And it looks like earlier event of hepatic failure or hepatic encephalopathy, index on the day of diagnosis, and then we excluded cases with prior chronic hepatic failure or hepatic cirrhosis or cases with liver transplant up to 7 days before index. This is just one example. So we took those algorithms, we implemented them across 10 different data sources that are ETL'd into the OMA common data model. You can see that they varied across geography, and most of them were claims data, but we have one electronic health record source and a few GPs data. Then we characterized the patients that met those definitions across these different data sources, and we reviewed that. So there's a tool in Odyssey called Cohort Diagnostics that allow you to generate a set of descriptive reports and visualize that data among a cohort of people in a given data source. And how is that useful is if you know the clinical idea you're trying to capture, you can look at the patterns of the patients you're identifying through the algorithm, and you try to see if that is consistent with what you expect. For example, for acute hepatic failure, this is one report that you can get out of Cohort Diagnostics, where here we're looking at the incidence rate of acute hepatic failure in 2 different data sources. And you can see, and it's you can see on the x-axis are the calendar year, and then stratified, the incidence is stratified by age and gender into different data sources. You can quickly realize that you actually don't have acute hepatic failure prior to 2015 in these data sources. And that's not because we did not have acute hepatic failure. Those are US-based data sources. In the US, that's just because those ICD-9 that were proposed didn't only have a very poor PPV, which we already know from the literature, but they clearly have a very low sensitivity, and they're really picking nobody. While once we get to the ICD-10 era, prior post 2015, you start seeing a occurrence of the event consistent with what you expect. So that gives you a quick non-validity of your phenotype in the pre-2015. Another report that you can get out of Cohort Diagnostic, that you can look at the distribution of the codes you included in your code list in the entry criteria among the people who met the definition. So you can see here that somewhere between 40 to 60% of the people were indexed on this code acute hepatic failure without coma, which is a very nice specific code in the ICD-10. But you also see that about 15 to 20% were indexed on hepatic failure and specified. This is a code that is more of a high level that does not distinguish between acute and chronic. What that means is maybe up to 15 to 20% of the people you're capturing is misclassified acute hepatic failure or chronic hepatic failure. Now, of course, you can remove that code and get down by 20%, but that also means that you maybe lost your sensitivity by 20% if those causes are true acute hepatic failure. So that gives you an idea of the boundaries of your measurement error depending on the decision you make in your code list. So that's a very nice tool that allows you to do that. You can also look at the entire characteristics, all the code distribution of the people who met the definition in different time periods related to the index. So here in acute hepatic failure, we learned from our tools that about 40 to 50% of the people had events related to encephalopathy around that time of index, which is good. That's something you expect. Some have jaundice, and some, about 30 to 50%, had elevated liver enzymes observed in the data. And that again give you some clinical validation that the cases you're detecting are what you expect and what you're looking for. Again, this is not perfect, but it gives you some idea of maybe potential errors or non-errors. Then the sixth step is where we selected cases. We looked at their patients' profiles again. This is structured data. This is not the full record with all the story of the patient. But what that gives you is, again, some validity, some clinical validity, or maybe identifying error in the phenotype. For example, in the case of acute hepatic failure, we found that there were some cases where patients went to the hospital, had an acute hepatic failure code, but within that same hospital visit, maybe a day or 2 days later, they had the code of chronic hepatic failure, which suggests that there might be some misclassification. In that case, we redefined our definition where we index on a hospital visit and require that the person does not have a chronic hepatic failure code within the same visit to kind of reduce some of that measurement error. Then in the last step, we estimated the measurement error using Phi Evaluator. And what Phi Evaluator does is that it's basically a predictive model that predicts the probability of having the event given the full set of a person data. And then you can use that as a gold standard and compare that to what your rule-based algorithm is doing. And what we see here for acute hepatic failure, we do pretty good in our PPV, somewhere between 70 to 90%, which we kind of expect looking at those records and how much clinical validity they have. However, we do a horrible job in sensitivity. This is really bad, somewhere between 19 to 29%. And that's probably, we know, because of the ICD-9 issue, because we're looking at the full set of the data, which include data prior to 2015. Okay, we estimated the measurement error across the entire set of our DME phenotypes, and you can see the performance of those algorithms. They varied by the DME. So we have some cases with really good performance, something like pancytopenia. You see some low sensitivity. I have to explain the graph you're looking at. So on the x-axis, you see the sensitivity. On the y-axis, you see the PPV. Each dot represents the estimate from a different data source. So you can see where the measurement error is as estimated by Phi evaluated. We also noticed that estimate sometimes varied by data source. So if you look at neutropenia, you see that wide range of sensitivity that went from 20% to 70% depend on the data source. Also variation in PPV, example aplastic anemia, where it went from 30% to 80%, depending on the data source. So I'll finish with our limitation. Obviously, the biggest limitation here, we did not do traditional case level adjudication to get the true PPV or somehow sensitivity, but we did what we can. And we noticed that variation by data source, and we know that this is always going to different also by time, depending with the differences in healthcare practices and uses of codes. And while we followed the structured process and we were trying to use objective measures to make decisions, we still find that we always land onto some subjective decisions that we make when we decide on which phenotype to use, and that might vary from one investigator to another, from one clinician to another. All those phenotypes are available on this Shiny app that you can all have access. It's the link is on the abstract. It's on the slides that you have access to, and you can look at them, take the code list, use them as you like. And I will just remind everybody that phenotypes and observational data is complex, multidimensional, and requires really exchange of knowledge across multiple disciplines. And thank you.
Diana Esposito, Moderna. Thank you so much. This was a really great presentation, and I do look forward to checking out the app. I'm curious if you can comment on the choice of using all data as opposed to restricting to 2015, at least for the US.
Yeah, absolutely. So we actually have the estimates for only 2015, and we see a much better sensitivity as we expect. But the idea here is we want to see how kind of rigor those phenotypes across multiple years. And because sometimes you take this and you implement it in data unknowing that you have changing codes. Sometimes in our case, when we run network studies, we have different vocabularies. So something might work perfect in an ICD-10 world, works horrible in a read vocabulary. So obviously if we're only thinking of a data for 2015 using only ICD-10, you'll have a very better measurement error, and that's what we estimate.
And if I can ask one more question, I know in this particular environment you weren't able to validate, but with the DME algorithms that you've developed, are you planning on moving that forward in other environments where you can, or not at this time?
So this work was for signal detection. So it's an internal process that we're following. So in that case, we won't be doing like traditional case validation, but obviously in all our work with like past studies, we do case validation, and we follow this process for all the phenotypes we do, and regardless of the use case. And in many cases, we end up doing some case adjudication if it's a regulatory request, and we were able to like kind of compare what we come up with, what we estimate out of the records. And sometimes we're very close, sometimes we're very far. It really depends. But yeah.
Hi, Aza. Thank you for a great presentation. Judy Morrow, Harvard Pilgrim Healthcare Institute. I wanted to ask a question. I noticed that oftentimes there is this thought like we have to run every algorithm on every database, but the databases are clearly quite different. And especially with acute hepatic failure, which you really expect to see in an emergency setting, the GP databases and the ones that are ambulatory only, I wonder about the reason to actually continue to progress to run things on those types of things. It sounds like it seems as though it will create a what do I want to say, potentially easily misconstrued picture of what's going on because the data are just not fit for purpose.
Yes, no, definitely. So I showed you the full universe of the data sources that we did our evaluation on. And that for some DMEs, it doesn't make sense to have GP data. For others, it totally does. Some things like anaphylaxis, you might want to be able to pick those ER visits or maybe GP visit that you captured at the time of event. So obviously now, I said so that work only included how we did the phenotype algorithm and we made them ready for our surveillance system. But our first surveillance system that we're working on doesn't just take those phenotypes as is and run it across all the data sources we have. We actually have a kind of 3-level gateways where the first step we look at the fitness for use of a data source for a particular analysis given the outcome measure we expect. So at that point, those GP data would fall for something like acute hepatic failure and wouldn't at all be considered for analysis. But I just kind of give you the body of data sources that are available and the body of phenotypes that we worked on. But definitely the match between the phenotype and the data source was something we look at the very first step of our process of running a surveillance system where we drop certain things that don't make sense.
Thank you, Aza. We're gonna move on to the next presentation just to keep up with time. So thank you. So next we have Hannah Johnson from St. George's University of London.
Hi everyone, my name's Hannah and I'm talking today about applying some new signal detection methods to the EuroMedicat data, namely the Bayesian confidence propagation neural network biclustering or BCPNN-BIC and the Bayesian confidence propagation neural network bicluster Bayesian hierarchical model or BBBHM. So just to let you know, I'm in receipt of a MRC-led studentship, which this work is funded by, and I have no other conflicts of interest. So onto the background. Signal detection methods are statistical processes that are commonly applied to spontaneous reporting datasets. And the way they work is effectively they can scan through all medication adverse reaction pairs simultaneously to identify areas where research should be focused. However, it's important to remember that signals are not evidence of a causal association. They are hypothesis-forming events, and they could be due to some sort of underlying confounding event. So we're interested particularly in teratogenic medications. These are medications that increase the risk of birth defects when taken during pregnancy. And actually, the majority of pregnant women take prescribed or over-the-counter medications during pregnancy, but the risk to the fetus for most medications is unknown. So we're using EuroMedicat. This is a unique database with information on both birth defects and the medications taken during the first trimester of pregnancy. They are population-based registry data from 15 different countries in Europe from 1995 to 2019. And so we wanted to look at these two new methods, the BCPNN-BIC and the BBBHM within this EuroMedicat dataset. These are methods that are new. They're both tailored to smaller datasets and they've been shown to be effective in simulation studies, but they're yet to be applied to a real dataset. So we wanted to compare signals from these methods and other commonly used signal detection methods within this dataset. So what did we do? We investigated harmful drug defect associations for 82 EuroCat anomaly subgroups to 3-digit, 4-digit, 5-digit, and 7-digit WHO ATC medication groups, comparing these 6 methods. So we used case malformed incidence ratios These are very similar to proportional reporting ratios, but because they're population-based registry data, they're closer to incidence ratios than reporting ratios. Sequential probability ratio testing, the traditional Bayesian confidence propagation neural network, Dumicel's gamma Poisson shrinker, and our two new methods. And in the interest of time, I'm not going to go over how each of these works, but if you are interested, then I'm around all day. Do come and have a chat. So going on to what we found, I'd like to make clear that these results are still preliminary. So we had data on just under 160,000 cases, and we looked at signals detected for each level of the WHO ATC. So for those who haven't come across it before, here's an example using the known teratogen isoretinin. So the 3-digit therapeutic group would contain all medications that are anti-acne preparations. So you're grouping together a large set of medications. Then at the 4-digit level, these would be all anti-acne medications for topical use. 5-digit level would be just retinoids used in acne. And then at the 7-digit level, it's just the specific medication isoretinin. So as you go down the groups, you're sort of pulling the groups apart. And what you end up with is more medications, so more medication defect pairs that you're investigating. And the results become more informative because you're looking at specific medications rather than sort of a big group of medications that doesn't really mean much. However, for each pair, you end up with less information. The data becomes sparser, which is obviously a problem. So here we have the proportion of signals identified by each method at each level of the WHO ATC as a percentage of overall medication defect pairs analysed, where there were at least 3 medication observations and they met the method's specific signal criteria. And hopefully what you can see is that the percentage of medication defect pairs is somewhat stable for most methods as you go down the groupings, except for the GPS, where it tends to find less signals as the data becomes more sparse. And overall, you can see that the SPRT and the GPS are finding far fewer signals compared to these other 4 methods at the top. This is the BCPNN BIC, the incidence ratios, the BCPNN and the BBHM, which are identifying the most signals. And that bottom line there in that group, the BBBHM, this identifies a lower proportion out of these 4 methods. And this would correspond to what we've seen in simulation studies, which suggest that actually it has a lower false positive rate than these other methods. So here's a Venn diagram. I realize this is there's a lot going on here. So I'm going to go through each bit. So the number in the middle is the number of signals identified by all of these methods. So there's quite a few there. Whereas the numbers on the outside are those that are being identified uniquely by that method. So this one here is the signals identified only by the BCPNN BIC. There's quite a few, and we'd expect there to be a relatively high proportion of false positives in that sort of group. Likewise up here, this is the incidence ratios. We have quite a few that are just found by there. And again, we'd expect a fair number of false positives. The interesting numbers are these. So this is the number of signals that were identified by those 4 top methods but missed by the GPS and the SPRT. This is the number missed by the SPRT and the number missed by the GPS, or in other words, found by all the other methods and not the GPS and the SPRT. And the pattern is very similar as you go down the WHO ATC groupings. But as you can see, as you go down, the number that are identified by all other methods but the SPRT and the GPS is increasing, which explains again that gap that we saw in the other figure. So we know from simulations that the GPS and the SPRT have a very low false positive rate, which is great, but they tend to miss true signals when the number of observations is low. And here we have a set of pairs that actually we would have really wanted to see. So this is valproic acid with cleft palate, carbamazepine with cleft palate and spina bifida. These are well-known teratogenic associations that we really would have wanted to be picked up by any signal detection method. It was not picked up by the GPS in this data. And likewise, at the bottom, we have insulin aspart with hypoplastic left heart and combined contraceptives with abdominal wall defects. These aren't necessarily causal associations, but they're documented associations that we would have potentially wanted to be picked up within this data set. And they were not detected by the GPS or the SBRT. They were detected by all of the other methods. So in conclusion, for smaller data sets with sparser data, the GPS and the SBRT may be missing signals of potential harm. The BCPNN BIC and the BBBHM are able to identify these signals within the Euromedicat data. And based on the patterns of signals found in this data and the simulation studies, this would suggest that the BBBHM has the lowest false positive rate, and therefore, in our case, may be preferred. So thank you very much.
Thank you for a very interesting presentation. Niklas Norén from the Uppsala Monitoring Center. So can you say a few words about the principles? I know you didn't have time to get into the details of the two new cluster or bi-cluster analysis methods, but can you say something about the principles of what you're trying to achieve and the added value they would bring over the sort of more basic disproportionality measures that you were comparing against?
Yes. So so if I explain how these methods work, that might help. So the BCPNN BIC, it's essentially the BCPNN with an added bi-cluster analysis. So what you do is choose a slightly lower cutoff, which dichotomizes the data, and then you apply a bi-clustering algorithm. And it's very basic. You basically say anything that was a signal with the traditional BCPNN criteria or meets a cluster definition, you consider as a signal. And it's quite intuitive, because what you're essentially saying is if a signal was on the borderline of being identified, but wouldn't have met the criteria for BCPNN, but it's behaving in the same way as a medication that's causing known adverse reactions, so it's behaving in a similar way as harmful medication, we can consider that as actually maybe that is something we'd want to look at. So that's sort of the added benefit. And actually in this analysis, we found it looked like quite a high false positive rate. But if you tweak the cutoff that you're using, that should be able to, you might be able to sort that out a little bit. The BBBHM is a further extension to that. So you're using the bi-clustering information to create hierarchies. So they're hierarchies that are generated from the data itself, rather than using sort of predefined hierarchies, which tend to be commonly used when you're applying Bayesian hierarchical model to this sort of data, like the WHO-ATC or MEDRA or things like that. So because it's generated from the data itself, you tend to get better hierarchies, and then you've also got the shrinkage that's applied from Bayesian models generally. So that's why the false positive rate is lower. Does that help?
That's very helpful. But just to test my understanding, by clustering, you mean you're looking both in the drug dimension and in the adverse event dimension, and you're looking for basically adjacent terms in.
Yes, exactly. So you're looking for essentially medications that are associated to two or more of the same anomalies, and vice versa. So two or more birth defects are associated with the same medications. Yeah.
Great, thank you.
Thank you very much, Hannah. With that, we'll welcome our next speaker, Lockwood Taylor from IQVIA.
right. Hi, good morning, everyone. My name is Locke Taylor. I'm with IQVIA. And now on behalf of our joint IQVIA Sanofi project team, I'm pleased to present the results of our exploratory analysis applying several different approaches of signal detection to patients on the anti-diabetes drug Lantus. Disclosures. So we know that recently we've seen increasing exploration of longitudinal electronic healthcare data, claims, EMRs, for purposes of signal detection. And one of the more popular approaches to this are the use of tree-based scan statistics, which leverages the tree-like structure of hierarchical data, such as an ICD-10 coding tree. We also have the ability to apply several different types of study designs aimed to reduce false positives due to minimization of confounding, as well as adjustment for multiple testing. Arguably, the biggest advantage to these tree-based scanner approaches is that there's no pre-specification of a potential signal. This really is hypothesis-free signal detection across an entire coding tree. So two designs that are commonly leveraged using tree-based scan statistics within tree scan software, the self-controlled tree temporal scan analysis and propensity score matched analysis. So given that these types of approaches are gaining more traction both in regulatory and in industry circles, and even in our own experience as a CRO, working with companies to implement these types of approaches into routine pharmacovigilance, some important questions have come up along the way that maybe have not so straightforward answers, including how do we interpret findings from tree scan, and should we expect to see the same types of signals that we see in, say, a disproportionality analysis. So with these questions top of mind, we had two objectives. The first was to run two separate study designs using tree-based scan statistics in a cohort of type 2 diabetes patients initiating an anti-diabetes medication. And second was to evaluate the performance from our tree-based scan statistics relative to that of a disproportionality analysis using spontaneous reporting data. So how did we do this? Well, first we built our cohorts. Our exposure cohort were new users of insulin glargine, and our comparator, just for purposes of the propensity score matched analysis, were new users of NPH insulin. Now, I appreciate that NPH insulin is not one of the more widely prescribed insulins this day and age. However, it was the insulin that patients were randomized to in the Lantus trials. These were new user cohorts. We had a 365-day exposure washout. We required 400 days of continuous enrollment prior to index prescription. Our study period was October 16 through June 2020. We limited our analysis to patients at least 18 years of age or older, as well as to type 2 diabetics, which is an important distinction there, I think. For our source of data, we used US Pharmetrics claims data, which is a large database of commercially insured patients in the United States. We did leverage the publicly available tree scan software for our two tree-based scan analyses. The first was a self-controlled tree temporal scan only in Lantus patients, looking in the first 90 days post-exposure. And here we compared the number of event clusters in all combinations of risk versus control windows in the first 90 days. We had a p less than 0.05 of alert threshold based on the maximum log likelihood ratio test. Our second analysis was a propensity score matched analysis where Lantus patients were matched 1-to-1 to NPH patients based on propensity score, and also had a 90-day risk period there. So we used incident events only with a 400-day event washout. We also excluded several categories of codes up front, as it was thought that these codes would be less meaningful if they did signal in this particular analysis. And these were largely code categories around pregnancy and birth defects. And finally, for both analyses, we assessed two outcome categories. We looked at ICD-10 codes in any care setting, so inpatient, outpatient, emergency room. And we looked at only ICD-10 codes in the inpatient and emergency room setting, thinking that maybe we would reduce some of the noise that can come up in outpatient claims data, as well as the possibility that these codes may more likely represent the more severe adverse events. So to address our second objective around comparator performance, we also ran a disproportionality analysis using data from FAERS for reports of insulin glargine, same population, same study period, and we applied 3 different thresholds to detect potential signals. We had an EBGM of greater than or equal to 4, an EBO5 greater than or equal to 2, as well as a composite metric. Finally, we compared the results from our tree scan analysis and our disproportionality analysis to a gold standard list of adverse events. This was based largely on events that are listed in the Lantus US product label and were supplemented with adverse events identified in the Sanofi PV database. All in, there were 93 true positive or gold standard adverse events that were mapped to 41 ICD-10 codes. For our negative controls, we randomly sampled 200 MEDRA codes and 200 ICD-10 codes, and then calculated our usual measures of accuracy. So what do we see? So here are select cohort characteristics before and after matching. I think the big take home here is that we had far fewer patients on NPH than on Lantus, which we expected going in. Even though our matched cohorts were reasonably well balanced, at the end of the day, we ended up matching less than 15% of our Lantus cohort to NPH patients for the propensity score matched analysis. All right, let's get into tree scan. So for our tree temporal scan analysis in any care setting, we observed 91 alerts. 3 of these were on our gold standard adverse event list. So this caught us off guard a bit. This is a lot of alerts. What could be going on here? Well, so keep in mind that this particular analysis included outpatient data from patients who have a lot of comorbidities, and it could be that our 400-day event washout just was not long enough to exclude all of these prevalent conditions. Second, there may be something about the timing of initiating or switching to Lantus and having follow-up visits with providers in the subsequent days. So perhaps you have poor glycemic control, you're switched to Lantus, and in the same week, you're following up with your ophthalmologist, your neurologist, rheumatologist, and we're seeing a bunch of codes there. It's not that these are associated with Lantus, but they are associated with poor glycemic control or advancing diabetes. Third, we did have a loss to follow-up of about 8% in this cohort, which may have inadvertently driven up a number of false positives in earlier windows due to patients who didn't have data in later windows. You'll see when we look at the tree temporal scan and limit only to inpatient and emergency room, we had 11 alerts. So the number of alerts decreases considerably in these care settings. And one of these was on our gold standard adverse event list. When we move on to our propensity score matched analysis, where we match Lantus patients to NPH patients, there were 7 alerts. None of these were on our list of gold standard events. And finally, when we limited to inpatient emergency room setting, there were no significant alerts detected. So finally, when we compare our results in tree scan using Pharmetrics data to our disproportionality in FAERS data against our gold standard event list, we see that the number of gold standard events that signaled was considerably higher from our FAERS analysis than in our tree scan analysis. And that while sensitivity was low across the board, regardless of the approach, both sensitivity and PPV were appreciably lower in our FAERS analysis than in our tree scan analysis. So what do we make of this? Clearly, we see big differences both in the numbers and types of signals between tree scan approaches, as well as between tree scan and our disproportionality analyses. So why? Well, we've used different data. We've used different study designs with different comparators. And as a result, we've really asked different scientific questions about the same product. So perhaps it's not surprising then that we would see very different signals emerge from these analyses. Regarding data with metrics claims and reporting data, clearly there are differences in what these underlying data represent. With respects to our tree scan analyses, we saw the impact of limiting our analyses to inpatient and emergency room settings only. We also saw our tree temporal scan as kind of a finicky analysis that seems to be sensitive to temporal patterns of care, which could particularly be problematic in a population of patients who have quite a bit of comorbidity. And then third, comparators. I don't think I think it's important to really look at what our comparators are in these analyses and how they may have driven the events. If you go back and think about it, our gold standard events, these were largely based off of clinical trials that occurred almost 30 years ago. And in our study, we had a self-controlled analysis, a propensity score matched analysis to NPH in the post-approval setting, and then a disproportionality analysis to all drugs and all events. So I think these key differences can certainly explain the variability in the types and the numbers of signals that we've seen. That said, I can appreciate that there's maybe some confusion or some frustration around how to interpret these results. That said, it seems to me that perhaps these results cannot be easily reconciled, and nor should they be reconciled. You've asked, again, very different questions. But it is important to keep in mind that all of these approaches do reflect patient experiences on medical products in the real world setting. And as a result, in our view, these methods and results should complement one another and contribute to our broader understanding of a product's evolving safety profile. So important limitations here. Clearly, this was just done on one product that was approved a long time ago. There are some issues around generalizability. I'll also add that I wouldn't put too much emphasis on our comparison to our gold standard adverse event list, given that these gold standard, that the origins of these gold standard adverse events are quite different than that of the origins of the signals from this analysis. I realize that's probably a topic, very different conversation, but still important to think about. And finally, we do believe there is a growing role for signal detection using both reporting data and longitudinal electronic healthcare data and routine pharmacovigilance. These approaches should be complementary and should be cautiously interpreted in light of underlying differences in data and study design. And with that, I want to acknowledge and thank my project team, and happy to answer any questions. Thanks for your time. Okay.
Bob Chen, Brian, collaboration again. Excuse me. Have others done similar kind of validation studies of tree scan approaches like you have, and have they had better outcomes?
So by better outcomes, do you mean?
Well, you seem to be kind of not so happy with.
Yeah, no, it's not that. I think the point is that we want to show that if you're going to incorporate these types of approaches into routine pharmacovigilance, it's important to see that you may get very different findings. And it's not that they're wrong. It's just they've asked different questions, and it's important to keep those questions in mind. That said, I believe there have been several other validation studies done in tree scan. They've all performed reasonably well. I'd have to defer to my colleague Judy to speak to those. But, yeah.
Hi, Chris Rowan, Core Data and Veritas Research Foundation. I'm just curious, why didn't you ask the same questions?
Well, how would you do it? I mean, right? So, I mean, I think that's the challenge, is that, you know, we had our gold standard events, which were based off well-controlled trials a long time ago in a very restricted setting. And here we are in 2023 trying to take some recent data and realizing that the patients on NPH today are probably look very different than our patients on NPH for the trials. We also know that the two approaches that we applied here are two of the more common approaches that can be applied in tree-based scan statistics. So I don't know, does that help answer your question?
Kind of.
Maybe you can take it after, just so that we stay on
happy to
schedule.
chat
Yeah.
after.
Thank you very much, Locke. So our last presenter, last but not least, is Jose Hernandez Munez from the FDA.
Okay, good morning, everyone. My name is Jose Hernandez. I am a senior pharmacist and pharmacoepidemiologist with the Sentinel Core team at the U.S. Food and Drug Administration. The title of my presentation is Investigating Potential Difference in the Safety Profiles of Biosimilars Relative to Originator or Reference Product using a tree-based scan statistics. This study, along with two others, was conducted as a method pilot to evaluate the potential integration of tree-based scan statistics into the FDA Pharmacovigilance toolbox. These are the co-authors that collaborated in the project from the Sentinel Operations Center and the FDA, and I want to acknowledge Malika Munkur from the FDA, who was the lead author in this project. These are my disclosures. Most important ones, the views expressed in this presentation represent those of the authors and do not necessarily represent the official views of the Food and Drug Administration. So biosimilars are biological products that are highly similar to FDA licensed biological reference products. Biosimilars must have no clinical meaningful difference from the reference product. So basically, this means that biosimilars should provide the same treatment benefits and have the same risk as a reference product. However, complex manufacturing process may lead to product variability, such as potential for difference in immunogenicity. Therefore, for post-marketing surveillance for difference in the safety profile between biosimilars and the reference product is critical. At the FDA, we conduct post-marketing surveillance using FAERS. However, there are some limitations with the use of FAERS for post-marketing surveillance of biosimilars. For example, adverse event reports for biosimilars may be inadvertently submitted under the reference product. With FAERS data, we are unable to control for multiple confounding variables in medically complex patients populations, such as those typically treated with biosimilars or biologics. Also with FAERS, we confront challenges in detecting difference in adverse events frequently between biosimilars and the reference product, particularly for commonly reported adverse events such as immunogenicity or lack of drug effect. For this pilot project, Sarcio was a selected biosimilar and Neupogen the corresponding reference product. Sarcio was approved on March 6, 2015, and Neupogen on February 20 of 1991. The two products are human granulocyte colony stimulating factor, approved for a variety of indications as listed in this slide. And as expected, the safety labeling is similar for both products. The analysis was conducted using 5 Sentinel data partners, and the study period included data from November 5th, 2016 through June 30th, 2021. We require eligible members to be aged 18 years or older and to be continuously enrolled in health plans with medical and drug coverage for at least 400 days prior to the index date, during which gaps in coverage of up to 45 days were allowed, as illustrated in the dark gray and gray areas in the chart. Only the first qualifying treatment dispensing or administration of Sarcio or Neupogen was evaluated. We define new use as no evidence of the index-defining exposure in the 365 days prior to the index date. We also excluded patients with use of the competitor exposure or to other filgrastim biosimilars and related biological products on index date or in the 365 days prior to the index date, as illustrated in the purple and red portions of the chart. Follow-up began on the day after the index dispensing and continued for 183 days or until the first occurrence of any of the following: death, disenrollment, end of the query period, end of data partner data, dispensing or administration of the opposite drug, or dispensing or administration of a related biological product, as illustrated in the orange, blue, and light blue portions of the chart. Among the 1-1 match Neupogen and Sarcio pairs, we evaluated the occurrence of health outcome of interest in the inpatient and emergency department settings using a hierarchical tree of ICD-10 diagnostic codes. In this study, we performed signal identification by monitoring non-pregnancy and non-cancer outcomes. For an outcome to be considered incident, there were required to be no outcomes with the same first 3 digits of the ICD-10 code in the prior 400 days in any setting. We evaluated incident health outcome of interest beginning 1 day after exposure initiation until the end of the exposure episode, provided that both members of the matched pair were still able to contribute at risk time for the outcome of interest, as illustrated in the green and light green portions of the chart. A sensitivity analysis was conducted, adding outcomes captured in the ambulatory care setting, where all other identification rules remain unchanged. Important to mention is that we assess age, gender, ethnicity, race, and I lost my cursor, into the propensity score model. And we made sure that we control for those during the analysis. Propensity score was calculated using these characteristics, and matching was performed using a 1-1 nearest neighbor matching without replacement on the probability scale using a caliper of 0.025. We'll have to use the cursor. Once the incident user cohorts were created, a propensity score matched, they were evaluated for the occurrence of outcome using TreeScan, the TreeScan software, to determine whether there were imbalance in outcome occurrence between the 2 groups that rose to the level of statistically significant. TreeScan is a data mining software that implements tree-based scan statistics to scan electronic health data that are grouped into hierarchical tree structure while automatically adjusting for multiple scenarios. Under the null hypothesis, my apologies for that. Under the null hypothesis, there's no position on the tree where outcomes are expected to be an imbalance. Yeah, I'm having issues with the mouse. I don't. Yeah, but that's for moving down. Okay. Okay. Let's see. My apologies for that. Okay. Yeah. So under the null hypothesis, there's no position on the tree where outcomes are expected to occur in a ratio other than the fixed match 1-to-1 of the cohort. We use unconditional Bernoulli tree-based scan statistics to calculate the log likelihood ratio for the 3rd to 6th level node of the outcome tree. Now I feel way better because I needed to mention that. While evaluating the results, an alert triage process was conducted to identify and differentiate expected signals, unexpected signals, and potential false positive signals. Through this process, we checked the labeled conditions, commonly report adverse reaction in the literature, and in patent-facing medical materials. We also checked for late coding of indicators or indications or infrequently coded comorbidities that are co-coded upon occurrence of another adverse event. In total, 43,009 1-1 match pairs were selected from the more than 218 million enrollees. Characteristic imbalance at baseline included ethnicity, calendar year of index date, history of cancer and recent chemotherapy, history of organ transplant and prescription drug utilization. After propensity score adjustment, 892,259 incident outcomes were observed, with around 50% occurring in the Sarcio group. Four unique alerts were observed, one in the primary analysis, which included, as I mentioned, the inpatient and the emergency department setting, and 3 in the sensitivity analysis, which included the inpatient, emergency department, and ambulatory care setting. However, after careful review from the study team, no regulatory action was pursued for reasons such as likely connection to factors other than a drug-event relationship. The only alert that was statistically significant in the model considering outcomes in the inpatient and emergency department setting occurred at the 4th node level, and it was polyarthritis unspecified. The total node outcomes for polyarthritis unspecified among Sarcio and Neupogen incident users was 32. However, the node outcome among Sarcio initiators was 28, an excess of 12 of the expected number of outcomes. This resulted in a relative risk of 1.75 and a p-value of 0.0174 using the unconditional Bernoulli look-likelihood ratio statistics. The 3 alerts that were statistically significant in the model containing outcomes in the inpatient, emergency department, and outpatient setting, or ambulatory care setting, I should say, were pain in the right leg, pain in the right lower leg. These 2 occur at the 6th node level, and other disorders of peripheral nervous system. This one occurred at the 3rd node level. The relative risks were 1.27, 1.30, and 1.32 respectively. In conclusion, 3scan identified few statistically significant imbalance or alerts for Sarcio. However, after clinical review of the alerts, the FDA determined that no further action was required. This analysis provides some reassurance regarding the safety profile of originator products and their biosimilars. The conducted signal identification study is subject to typical limitations common to observational study. In addition, signal identification by nature is designed for a broad screening without a specific confounding control for targeted outcomes. Also, we noted some temporal substitution effects leaning towards biosimilar substitution over the query period. Therefore, we recommend the inclusion of calendar year of index date in similar analysis to adjust for this phenomena. This concludes my presentation. Any question? Yeah, hopefully I will be able to make up for some clarity because of the mouse issue with questions. So if any questions arise, please, now is the moment. Yes.
Sorry, could you go to the mic? Just because, thank you.
I was wondering if you could comment on the time frame for the washout for the outcome. Is the 1 year look back, but I was wondering, for example, if people could have recurrent infections, if that wouldn't be important to capture as well.
Meaning you're talking about the washout period or
you mentioned for the event, people had to have a new event without having the same diagnosis 1 year prior.
Exactly.
And in my mind, it came like, oh, so you're excluding if patients had repeated severe infections. So yeah. Anyways, that's just one.
That's a good point. So you're saying basically, to rephrase the question, is because we're excluding an outcome if it occurred 400 days before the actual outcome occurred, then we're probably eliminating recurrence events such as infections. That's your point, is that right?
Yes. Yeah. So I'm thinking if people, in some ways, you could have higher frequency of the specific events that could be important.
Yeah.
And if you have thought about changing this testing in a sensitivity analysis, this time frame for the outcome.
Excellent recommendation, and thank you for clarifying that. So yes, we are identifying the event. However, we are not identifying the intensity of the event, which is the point that you're trying to make, and that's a great idea, and we will consider for future analysis as a potential, value for intensity of the signal that we are identifying. However, keep in mind that this method is solely used for signal identification, and after being evaluated by the clinical staff, then we decide if we need to raise it to the level of a NIS or newly identified safety signal, and then proceed with a more established signal evaluation study. But excellent point. Thank you.
Kevin Haynes, J&J. So how much of this gets corrected for all the multiple testing? I mean, getting these signals, I guess, also to the other one. I mean, I'm definitely gonna start injecting this in my left leg, not my right, because there was no signal. But like, where does that come into this play with regards to the massive multiple testing here?
Yeah, it's a one-sided multiple site testing, and it adjusts for all the testing that you're conducting, all the comparisons depending on the tree size. Because as you know, and I don't know if it was clear on my presentation or log presentation, we check each node of the tree to make sure that there is an imbalance or no imbalance on each one of those outcomes. Does that answer the question? Perfect.
Well, we are at 10 o'clock, so I will release everyone. There is a break. There is coffee and tea, I believe, downstairs. There might be some upstairs. But I want to thank all the speakers today for very interesting presentations. Thank you very much.